Okay, so picking up from the last video, I'm going to talk about the laws of inheritance. So first off, let's talk about alleles. So when you think of an allele, think of like the flavor of the gene. So it's kind of like the expression or the trait that comes from the gene. So think about ice cream flavors. Ice cream is the gene, and the different flavors would be alleles. So Mendel only worked with 2 alleles at a time, but you can have more than one allele. Many different alleles go into things like our skin color and our hair color. That's why we see so many variants in those. So in the book, you see it talks about phenotype, genotype. It's got a nice little chart here giving you an example of what a genotype looks like versus what a phenotype looks like. So the phenotype are the physical traits or characteristics that you actually see. So we would see the yellow color, the green color. Versus the genotype, we're talking about what 2 types of genes were put together. So we're going to talk about what these mean. All right. So we have homozygous dominant, homozygous recessive, and heterozygous that you have to know. So let's see. Homozygous recessive, homozygous dominant, and heterozygous. Alright, homo means same, hetero means different. That helps. Recessive, remember we're talking about the lowercase letter. Dominant, we're talking about the uppercase letter. So if the gene is being represented as dominant capital R, recessive lowercase little r, that's kind of where we're getting that from. So if these were the 2 that I'm using, so if this is the dominant and this is the recessive, and I'm trying to put it in these forms. So homozygous recessive means they're going to be the same. So by same, I mean 2 capitals or 2 lowercases. So which one? So that's when you have to look at this recessive and dominant. So it's the same. So they're both going to either be capital or they're both going to either be lowercase. And in this case, it's recessive, which lets us know they're both going to be lowercase. So it'll be R, R. This one, homo, same. So we know they're both going to be capital or they're both going to be lowercase, and we look here and it's dominant. Dominant is the capital R. And usually the capital letter is the dominant. So they're both going to be the same, 2 capital R's. Heterozygous is the easy one. Heterozygous means you got one of each. So our only option is, it's pretty straightforward. So these are considered the genotypes. This is how you write a genotype. You write the homozygous recessive, homozygous dominant, or heterozygous forms. So when we do Punnett squares, when you get your outcomes and you see things in your Punnett square like this or like this or like this, you need to be able to tell, okay, that means heterozygous, that means homozygous dominant, or that means homozygous recessive. That's how you read the Punnett square. So the phenotype for this same situation, so let's say the capital R means green, so green is dominant, the dominant color flower, and lowercase r is the yellow, which is the recessive colored flower. The green is dominant to the yellow. So in this case, obviously, this one's going to be green. In this case, we don't have anything but yellow, so it's going to be yellow because there was no green to dominate the yellow. It's just 2 recessives. And then here we've got a little bit of green and a little bit of yellow. So when you put those together, the green is going to dominate. So in this case, we've got a yellow flower. This one is going to be a green flower. And this one is going to be a green flower. Okay? Make sense? Okay. All right. Moving on. So, again, there's a chart here. This is page 103, if that helps. Again, some more information here. on genotypes, phenotypes, a comparison of the two to help you understand. So now we're going to talk about the law of dominance. The law of dominance states that in a heterozygote, so what's a heterozygote? So that was like this, right? This is the law of dominance. One trait will conceal the presence of another trait for the same characteristics. Well, that's what we just saw, right? Because when we combine the capital R, which is green, and the recessive yellow, the flower turned out green, right? So we just saw that. So that's what law is making this possible for us to do these Punnett squares and to make it come out right. And in the book, you'll see where it talks about the violet was dominant to the white in Mendel's experiments. So that allows us to make these wonderful Punnett squares that everybody so loves. Gotta keep your humor. Monohybrid cross, or a dihybrid cross. And both of these are types of Punnett squares that we will make. So when we do mono, we're talking about one type of trait comparison versus di, we're talking about two types of traits to compare. Dihybrids are a little trickier. Mono, pretty straightforward. So the mono, they only differ by one characteristic. And so anytime you have a monohybrid cross, like in the last video, the one that we did, that would be considered monohybrid. Remember, we had the 3-to-1 phenotypic ratio. And what we did in the last video was a Punnett square, so if that helps. So there's different ways we could write it. So because we had the 3-to-1 ratio, so the 3 dominant to the 1 recessive, we could also say in percentages. So this would be 75% versus this being 25%. So there's a 75% chance of having in the last video we did purple and white, so there's a 75% chance of having a purple flower and a 25% chance of having a white flower. And you'll see that on page 104. It's kind of talking about different ways to write it, but the main thing you need to know is that 3-to-1 ratio, and how to, manipulate it and change it to this form if necessary on the test. But that's doing monohybrid crosses. So the monohybrid cross is when we do something that's going to have 4 outcomes. Right? We have, just make something up, right? This and this and this. So all of those would be heterozygous if we work that out. So if I was going to write the genotype for this, it would be 100% heterozygous. And if I was going to write, so that's the genotype. And if I was going to write the phenotype for this, and R is green, they would be all green. So genotype, phenotype. Okay. All right, so on page, I'm sorry, let me not skip law of segregation. So a lot of students struggle with differentiating the laws. There's 3 laws. So we've already talked about the law of dominance. So now we're doing the law of segregation. And that just states that, if you remember from, I think it's in chapter 6 or 7, when we talked about meiosis, I think it's chapter 7. Remember in meiosis 1, we had the crossing over of gametes. So when the male and the female came together in the process of forming the zygote, their genes kind of crossed over to allow for that recombinant DNA to occur, because it increases the outcomes and the different variations. So we like biological diversity, and that's what it does, so we favor that. So this law of segregation is just another way of saying that in terms of genetics. He's talking about the genes are equally likely to go into the offspring. So the genes from mom and the genes from dad have an equal chance of going into the offspring. That's really all that it's getting at. So that process that happened in meiosis, he's saying mom and dad both stand an equal chance because of that crossing over of the genetic information. Okay? Pretty straightforward. Alright, top of 105, talking about a test cross. So a test cross is a technique used by plant animal breeders. It's basically if you take and you cross the dominant organism with a homozygous recessive. So, and it shows you what happens. So I'll work it out. Gametes from the parent of the unknown genotype versus gametes from the recessive parent. So here's two examples. So if I was doing a test cross and I have a homozygous dominant with a homozygous recessive. These are capital, these are lowercase. And I cross them. I'll do my little Punnett square. So I put capital, capital for this guy. And for this guy, I'm doing them on the side, lowercase, lowercase. Then I take whatever's hitting that square and put it in there. Capital first, lowercase. Capital first, lowercase. Capital first, lowercase. Capital first, lowercase. Okay, so they're all the same, right? So that was like the one we just did. It gave us 100% hetero outcomes, right? Because all of these are heterozygous, a capital and a lowercase. So they all would possess this dominant color, whatever it was. Okay? So now I'm going to take the outcome of that, which was the heterozygote, and I'm going to cross it with a homozygous recessive. So do the same thing. Hit up my square. Whoop. I'm trying to look at my paper and the phone at the same time. Sorry. So I'm going to put this guy on top. Sorry. He's capital. He's capital, sorry. And these are both lowercase. So capital, lowercase, 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 capital, lowercase, lowercase, lowercase. Alright, so let's write out the genotype and phenotype. So the genotype for this, let's see, we've got 2 heterozygous, so that's 50%. And we've got 2 homozygous recessive, so that's also 50% homozygous recessive. And instead of 50%, you could write 2 out of 4 and 2 out of 4, right? Because you got 2 out of 4. Or you could simplify that to 1 out of 2. So, either way. The phenotype for this, so these would take on the dominant phenotype, right? They have the dominant. So they would be whatever this Y trait is. So we'll say Y is red. So they would be 50% red, or 2 out of 4 again, or they would be, these are both homozygous recessive, so let's say the recessive is pink, so they would both be pink, so that'd be 50% pink. So the test cross brings you to this point of a 50-50 outcome. So the test cross consisted of both these parts. You're taking a homozygous dominant and a homozygous recessive, crossing it. Then you're taking that outcome, that heterozygote, and you're crossing it with a homozygous recessive, and it gives you this 50-50 outcome. Okay? Alright, last law. Law of independent assortment. And I'm about to start a new video because I don't want to lose what I have. So we'll start the next video here.